Gram stain was discovered by Hans Christian Gram in 1884. It is used to differentiate bacteria based on their cell wall composition and is essential for bacterial identification and classification. There are two main groups of bacteria identified using Gram staining. Gram-positive bacteria appear purple. Gram-negative bacteria appear pink. This difference is due to the cell wall structure. Gram-positive bacteria have a thick peptidoglycan layer that retains the crystal violet stain, whereas Gram-negative bacteria have a thin peptidoglycan layer with an outer membrane, so they lose the primary stain and take up the counter stain. Objectives: differentiate bacterial species into Gram-positive and Gram-negative groups using the Gram staining technique. To understand the role of different reagents, crystal violet, Gram's iodine, alcohol, and safranin. Equipment: sterile inoculating loop or needle, crystal violet stain, Gram's iodine, 95% ethyl alcohol, safranin stain, bacterial cultures, distilled water, Bunsen burner, clean glass slides, compound microscope. Procedure: label one end of a clean dry glass slide. Place a small drop of distilled water at the center of the slide. Using a sterile inoculating needle, aseptically pick a small amount of bacterial culture and mix it into the drop of water. Spread evenly to form a thin smear. Allow the smear to air dry completely. Heat fix by passing the slide through a Bunsen burner flame 3 times. Flood the smear with crystal violet and let it sit for 30 seconds. Crystal violet, a positively charged dye, penetrates the bacterial cell wall and cytoplasm and binds to negatively charged components, staining all bacteria purple. Rinse gently with tap water for 10 seconds. Add Gram's iodine and leave for 1 minute. Gram's iodine forms a complex with crystal violet, making it less soluble, so Gram-positive bacteria retain the dye during decolorization. Wash with tap water for 5 to 10 seconds. Decolorize with 95% ethyl alcohol dropwise until runoff is clear. Ethyl alcohol dissolves the outer membrane of Gram-negative bacteria and washes out the crystal violet iodine complex, while Gram-positive bacteria remain purple due to their thick peptidoglycan layer, allowing clear differentiation between the two types. Rinse with tap water for 5 to 10 seconds. Flood with safranin and leave for 1 minute. Safranin stains Gram-negative bacteria pink because they were decolorized by ethanol, while Gram-positive bacteria remain purple due to the crystal violet iodine complex trapped in their thick peptidoglycan layer, giving a clear color contrast for differentiation under the microscope. Rinse gently with tap water for 5 to 10 seconds. Air dry without blotting. Place the slide on the microscope stage. Start with low magnification to locate the smear. Move to higher magnification to focus the area. Place a drop of immersion oil on the smear. Observe using the 100 times oil immersion objective. Gram-positive bacteria appear purple. Gram-negative bacteria appear pink.